We left off on this slide in the previous presentation, talking about or explaining the reason why we cannot just go straight from the energy in sunlight to making some molecule that can be used to provide energy for the cell. And that, again, was because that would be too much energy for one single molecule to hold. So instead, we have a number of steps that gradually decrease the energy or share the energy from a photon of sunlight. So this image is an illustration or an abstraction of that concept, where we see many small steps down between the high energy electron at the top left and the waste molecules at the bottom right. And then in between, we have the energy being contributed or donated to different reactions and different molecules, which all have their own function. And so again, the idea is that this energy, all this energy is held in an electron, and we can't give it all to one molecule or one reaction, because that would be essentially an explosive reaction. Instead, we go through a series of smaller reactions with smaller amounts of energy and give a little bit of the electron's energy away at a time, which then is going to, all these different reactions are going to contribute in the end to the construction of molecules that can hold a reasonable amount of energy for use in either the cell or in the storage of energy in the body or the organism, depending on what we're talking about. So again, we're going from high energy to low energy, and then each of these steps in between looks kind of similar to something we talked about before, which is a reaction coordinate. So essentially we have a mini reaction coordinate at each of those steps, where we go again from some energy level. The energy increases temporarily because we have that transition state, and then as energy is released for that particular reaction, we have the energy of the products dropping off lower than the reactants. So this is a diagram of all of the important things that happen in photosynthesis at the molecular level. And it's summarized in about 3 steps here, but this is packed with information. So let's take a moment, take a step back, and look at it one piece at a time. So on the left here, we see the organelle where photosynthesis occurs. And this is the chloroplast. So you've probably heard of chloroplast before. They are green because of the chlorophyll, which is that green pigment that is in the inner membrane of the chloroplast. So this entire thing is the chloroplast. And then the chlorophyll is only in the inner membrane here, because remember, chloroplasts and mitochondria have double membranes because we believe that they were engulfed by eukaryotes back long ago when they were free-living prokaryotes. So you have the prokaryotic membrane and the eukaryotic membrane. And a lot of the reactions are actually going to occur in the inner membrane of these organelles, of the chloroplast, and when we get to it, the mitochondria. All right, so as we mentioned, the chlorophyll is in this inner membrane, and it is important because the chlorophyll are the pigments that are actually going to help to absorb the light or the photon. So we have the beginning of photosynthesis here, where we get a photon of light, which is absorbed by something called an antenna complex. This antenna complex has a couple of different jobs. So the antenna complex is going to, 1, absorb the light, and 2, it's going to actually split a water molecule. Now, what does that mean? Well, the water molecule, the H2O, or the hydrogen the two hydrogen with the oxygen is going to be split, and one of the electrons is going to be taken from that former water molecule, and that electron is what actually grabs the photon of light, or what receives the photon of light. Now, electrons, if you think back to chemistry or the beginning of the semester, electrons can exist at different energy states. And you could have high energy electrons or low energy electrons. They could move from different energy levels, I guess you could call them, or categories. And so this electron right now is high energy because it absorbed the energy from that photon of light. And now it's going to go through a couple of different systems of the cell and donate energy to these different reactions. So we see the electron is interacting with various proteins, and it's donating energy. It's giving off a little bit of its energy every time it interacts with these proteins. So again, we're seeing these steps that are occurring, giving away a little bit of that photon of energy, a fraction of the photon of energy at a time. The really key part here is that some of the proteins that the electron interacts with their job is that they are proton pumps, which just means that they move hydrogen across the membrane, and positively charged hydrogens. They move them across the membrane. All of these transmembrane proteins, these proton pumps, are oriented in the same way, so they're all pointing in the same direction, moving hydrogen in the same direction. And so if they're all working together, all pushing hydrogen through to one side of the membrane, that means we're going to get a higher concentration of protons on this side of the membrane, right? Well, that means that we have a concentration gradient going on now, or in other words, the concentration is different on each side of the membrane. It's higher on one side than the other. And the protons are going to go back through to try and go towards equilibrium through this other channel. And this channel is a protein called ATP synthase. And if you were to guess, you might guess that from its name, it synthesizes ATP. But it only does this if protons are moving through to the other side of the membrane. So that is a favorable transition or movement because the protons are going from high concentration to lower concentration. And so as they move, the ATP synthase kind of piggybacks on their movement from high to low, and it uses that to make ATP. Now, I'll talk about this in more detail in another video, but this, I just want to give you the whole context, and this is the whole context of photosynthesis. Again, we're talking about a bunch of reactions that are occurring in the membrane, and specifically in the thylakoid membrane, which are, the thylakoids are these inner membrane pockets or areas in the chloroplast. And so we've talked about a bunch of different steps that happen in the light energy or the light reactions of photosynthesis. And the light reactions occur first. So you have to have the light reactions before you have the dark reactions. You can break down the two different parts of photosynthesis into the light and the dark reactions, or the photo and the synthesis, or the photon driven reactions, and then the synthesis reactions. And we'll talk about that in a moment. So again, you can kind of summarize this in one sentence. The reactions in light portion of photosynthesis include the absorption of photons, the pumping of protons, and ATP synthesis. Now, of course, you could talk about this in much more detail. We're not going to talk about all the details, but we do want to remember a few. So we want to remember that, again, that electron comes from water. Water is split in the very first step, and so that electron from water is what actually gets the energy from the photon, picks it up and carries it around through these other areas of the membrane. Then the electron is passed between enzymes, and some of its energy is used to perform a reaction. And of course, the electron isn't like running around on its own. These proteins are transferring the electron to the next protein in the sequence. So going from one to the next, and it has to go in sequence. And the proteins themselves are helping that to happen. One of the things that we did not see here in this small diagram is the synthesis of NADPH. So NADPH is what is produced by one of these enzymes in this system. Again, a membrane protein that is an enzyme, and it uses energy from an electron that has some of the energy from a photon, and it makes NADPH from NADP. So in other words, it just adds hydrogen to NADP. And NADPH is important because it is a molecule that's going to be used later on. And then, of course, as we said, ATP is made in the last steps of photosynthesis, in the light reactions at least. I also wanted just with the big picture to explain, you might remember this from studying photosynthesis previously. Some individual organisms have photosystem 2 and photosystem 1 as well. So some types of plants have both of these, a lot of types of plants. Some algae. Bacteria only have one of these photosystems. And that's why it's backwards. When some species has both photosystem 1 and photosystem 2, photosystem 2 actually occurs first. And again, that's just because it was discovered second, because we studied photosystem 1 first. I think it was found in bacteria first, and so we knew about it first. It's called photosystem 1. So again, if you are looking at a plant species, it probably has both of these photosystems, and photosystem 2 comes first. And really, it's just a repeat of what happens here. So we get an electron, it goes through, and you get the pumping of protons, synthesis of ATP. But this electron can actually be passed and then re-energized. This is showing both photosystems. As we said, we get, again, the electron gets the photon of energy, and then it's being passed around, and then it gets re-energized for photosystem 1. So that was photosystem 2, this is photosystem 1. So the thing to remember here is that the electron gets energized twice, but we only use one molecule of water, which is at the very beginning because we're using the same electron. So as we see here, we have 2 molecules of water, and we release 2 oxygens in the form of O2, and we released 4 protons, and then we have 1 electron, which is taken and given that light energy, and that same electron is going all the way through, and then it's re-energized in the second antenna complex during photosystem 1, and then we see that it progresses on to donate energy to these other enzymes. And I call this the end, but actually it's happening or it's happening at the same time because the electron doesn't actually bind to ATP synthase at the end. So keep that in mind. ATP synthase is using the proton gradient. So while the proton gradient has to build up, And that's being done by these proteins that are using the energy from the electron. So they need to get the electron first. But once that proton gradient is built up, it's maintained constantly. And so as long as that is in play, that there's a higher concentration of protons on this side of the membrane, then ATP synthase is always going. So you can have the absorption of a photon as simultaneously with the working of ATP synthase.